start with the next speaker. I will make his introduction. Okay. It's about the Dr. Peter Groenendijk. He comes from State University of Campinas, Unicamp in Campinas, Brazil. About his biography. The Dr. Peter Groenendijk is an assistant professor at the State University of Campinas, Unicamp, Brazil. Is a distinguished scientist in the field of tropical dendro ecology. With academic credential from Utrecht University and Wageningen University, the Dr. Groenendijk's expertise and leadership have made significant contribution to the understanding of tropical forests. His role at the Unicamp placed him at the forefront of research in forest ecology, where he continues to influence the scientific community with his profound knowledge and dedication to environmental studies. The topic that he will speak in this conference is about tropical tree networks, research advances, climate growth signals, and drought responses. So you are welcome to this conference, the Dr. Peter Groenendijk. We can see your presentation.
Yes, I was looking at the ways of unmuting my audio. Can you hear me? Can you see me? Can you see my pointer?
Yes.
Cool, nice. And is my audio?
We can hear you too.
Cool, very good. So thank you very much, Kelly. It's a really nice opportunity to be here at the first Ecuadendro. And then do a little contribution talking about tropical tree rings. I think there were quite some nice things about Neil Peterson's presentation that are quite relevant for tropical tree rings, because it's the stuff that he showed about Cofetia and all these problems. For us, it's basically everyday life and getting to know your species and your anatomy and what's a ring and what's not a ring and how they fit and all of these things. So I'm not going to continue on the theme, but it was nice to hear about it. And I think it's tough for a nice discussion. I think we could use a paradigm shift in what is dendrochronology versus dendroecology and how these how do we say it, how the metrics compare. So sometimes I feel that the tropical stuff, or this more difficult terrain ecological stuff, is being judged with the same optics and metrics as dendrochronological stuff. And I think we could go and move away a bit from that. But that's maybe a theme for another conversation. So I'll be presenting results today of our tropical tree network. So in the last couple of years, 6 years, I think, we've been contacting researchers all over the world to get them to collaborate with us and build a network of tropical tree chronologies. And with that, we can explore how the research has been advancing. We've been looking at large-scale climate growth signals in the tropics, and also recently to drought responses. And I'll try to jam 3 major fronts and major presentations, major research works in one presentation. So we all know that tropical forest ecosystems are really important. Conditions there allow the system to process a lot of carbon, and they do transpire a lot of water. So they are really important for the global carbon and water cycles. And they dominate carbon fluxes over land. So the dynamics of tree growth or vegetation growth in the tropics drives the dynamics of global carbon fluxes. But the responses of these trees to climate change are still uncertain. and we need a better assessment of the climate sensitivity of tropical tree growth so that we can better inform, for instance, global models that take this sensitivity into account when projecting future scenarios of climate change. And drought in these ecosystems, in tropical ecosystems, they do reduce gross primary productivity, GPP, and with that, they reduce the carbon stocks in these ecosystems as well. So along with the climatic sensitivity, it's important to understand how drought affects growth and carbon sink potential of these systems. And tropical drought may reduce the long-term carbon sequestration and carbon storage of these forests, and most of it will be, or a large part will be because of reducing woody productivity, another part because of mortality patterns. And an assessment pantropically of drought effects on tree growth is still lacking and will be really important to improve our understanding of these responses. And we believe that tree rings may help. So we need a pantropical growth sensitivity and a drought effect analysis, and tree rings can help answer this question. But if we look at the ITRDB, so this is a Fleury-Babst paper from 2019. If we look at what is in the International Tree Ring Data Bank, there's almost no work being done in the tropics. And at the same time, we know that there are many people doing this explorative work in the tropics, sometimes against the odds and with a lot of effort. We are building this, but it's still a bit under the radar. There's not a culture of sharing these data to international databases. And I think from like a more developed country perspective, there's an idea that there are no trees in the tropics anyway. So there are many reasons why these are under the radar. And what we did is then assemble these. We contacted researchers all over the world. So between me, Peter Zuidema, Valérie Drouet, and Fleury-Babst, and we assembled this large network with now about 500 chronologies that have been published and are about to be published. And we have a website, the Tropical Tree Network. Already a paper out, and we're working on two major papers now still. And we have these 500 chronologies from 173 collaborators all over the tropics, and also a lot of people in temperate regions that have collaborated. So it's a major effort. And what I'm going to be presenting today are three main fronts that we're working on. One is a more general quantitative review of the development's potential and where there are research gaps in tropical dendrochronology. And this is a paper that is currently under review in Veterinary Science Reviews. Then I will talk a bit about the main patterns of tree growth sensitivity to precipitation and temperature. That's a publication we have from 2 years ago, almost 3 years ago now. And then another one that we're preparing, or it's under review, on the tree growth responses to drought events. So I'll start with the quantitative review. And we did a lot of things, and what I'm going to show here is the developments over time, where we can identify potential for tree ring work in the tropics and also for building century-long chronologies. And then I'll talk a bit about representativity, knowledge gaps, and research needs. So doing tropical tree ring work is hard. It often happens in isolated locations. So this, for instance, here in the lower left is fieldwork from my PhD in Cameroon, Central Africa, and it took quite some efforts to get these samples. I think I killed 3 4x4 pickup trucks for my PhD. And often the wood is hard, so we use these mechanized gasoline-powered boarders to get the samples out. or we have to do very hard coring, and it is then limited to species that can be cored, which is not always the case. So there are many challenges, not only for the hardness of the wood, but also logistical and financial to get this work done. But there's quite a lot of work out there. We've been looking at literature. There's some reviews by Rob Brinen, for instance, but also some work, more recent work also in South America. And we can identify at least or about 500 species with proven annual rings. So that's not necessarily species that have chronologies. So for instance, the species I studied for my PhD, I was unable to build chronologies. The cross dating was really messy. The rings were very complacent. But with 14C dating, we could identify that these rings are formed annually with maybe a few dating mistakes, but they are basically annually. So that's a species that forms annual rings, but does not allow to build chronologies. And one of the fantastic things about tropical dendro is that there's so many anatomical markers for what is a ring. So we could have parenchyma bands, but also alternating densities of vessels versus parenchyma, wood density variations. We also have semi-ring porous species like Tectona, Cedrela, Melia that form these large vessels at the beginning of the year. So it's really important to learn the anatomy of your species and really get the feeling what is a ring and what is not a ring. And what we found is that over time, so now we're talking about chronologies, there's been a strong increase in the number of chronologies that have been published. So it's almost an exponential growth in South America. It's a steadier growth in Asia and in Africa as well, with a lot of work on gymnosperms, also some work on angiosperms. In the Americas, they go more or less hand in hand. And if we look at the number of species, we see that the growth is resembles even more an exponential growth. So there apparently is a big potential of identifying species that form rings, especially in angiosperms. And this potential does not appear to be slowing down. So there's quite a lot of work to be done. And if we imagine that there's about 12 to 16,000 species in the Amazon alone, 3 species, 150 in all the Americas, or about 60 species in all the Americas is still really underrepresentative of what there is to be studied. We then took all these species that form rings, and we built species distribution models for each of these species. So you get a probability of the species occurring in each cell, each pixel of your map. And then we did a probability sum. So we summed all of these probabilities for each pixel, and then you can get an accumulated probability of species occurring here. And at some areas, we do find that we could have up to 25 species that have a proven potential to form annual rings. So that's a very substantial number, especially if we take, for instance, Neil Peterson's forest that he talked about that has 8 species, and it's considered a mixed forest. We have some areas with at least 24 species that have potential of this, I don't know, 400 or 500 species that occur there. So then we wanted to know where can we build longer chronologies, so 200, 250 year long chronologies, and that can be used for climate reconstructions. And we find that the chronologies, they have varying lengths. So for angiosperms, they go up to 200 years. Gymnosperms, they get much older, so on maybe around 350, 400 years. And the main factor determining the length of the chronology is mean annual temperature. So in areas that are very cold, so with low mean annual temperatures, we do get longer chronologies. And gymnosperms, on average, they also produce longer chronologies than angiosperms. And then you might be asking yourselves, well, this is about tropical chronology. How can we be talking about such low temperatures. But we do have very high mountains in the tropics. So in the Andes, for instance, where we do get very low temperatures as well. So it is not a tropical climate, but it is in the tropics and it's tropical tree growth. It depends a bit on how we define the tropics then. If we then take the species distribution models and we put in chronology length instead of presence, we can maybe give an estimate of areas that have the potential of building these longer chronologies. And we do have several areas that have this potential. And there's also quite some examples of studies that have produced these 200-year chronologies in these areas. So we believe the potential is not only in the number of species, but also that we can use tree rings to build these longer reconstructions. Then we have this network. It does have some spatial biases. So we see a lot of work here done in Central America, a lot of work done in the Himalayas. And we wanted to know how well the climate of the tropics is represented in by our network. And we just compared the climate envelope of our network with the climate envelope of forested areas in the tropics, so that have more than 10% of tree cover. And we can see that there are some biases. So this is for South America, and we see that the network is biased towards drier and colder areas. So we have mean annual precipitation on the x-axis, mean temperature on the y-axis. So the median of the network would be around 800, 900 millimeters, and of all the Americas forested areas will be around maybe 1200. So we have this bias towards drier and colder areas, and that has to do with all these studies in Mexico and in the Andes as well. They tend to be there's biases there that pull on these distributions. If we look for all the areas, we see that there's not so much done in Africa in general, although the climate envelope seems to be rather well represented. There's just too few studies there in general, and we see also some strong biases in Asia, especially towards colder areas. That's the Himalayas in our data. So tropical dendrochronology, so the conclusion of the first part is that there's a fast development in the Americas, few studies in Africa. We can build long chronologies with many species producing rings and many places with potentials to build 200-year-old chronologies. So for instance, this is a PEñA-Ecuadendro. It's a legume, tropical legume, and this tree was 230 years old. So we could potentially be building long chronologies in the southern Amazon. But there are some representative issues and biases that we should work on. The focus has been a lot on gymnosperms and high elevation sites, and we should do a lot of effort to go towards tropical humid areas, and especially in the African continent, there's a lot to be developed there still. Cool, that was the first part. Second part is about the main patterns of tree growth sensitivity to precipitation and temperature. And we basically wanted to know whether there are large clusters of climate responses in tropical trees, and how sensitive is tropical tree growth to precipitation and temperature, especially on a seasonal level. Okay, this work has been published, and there's a lot more there than only these two questions. So please go and check it out. So the clusters of growth responses, clustering is a bit of a magical, maybe mythical thing that you can choose many. You can choose out of about 30 methods to do your clustering, and I think methods do have effect on your results. And we have already used self-organized maps, which are considered robust when compared to other methods. And we did clustering on the temperature and precipitation responses. So we took the current year temperature and current year precipitation data together with the previous year temperature and precipitation data. And then we looked at monthly correlation coefficients. So we do have the precipitation response in blue and temperature responses here. And in this example, we do have a positive precipitation response, especially in the beginning of the growing season. So on this transition between dry and wet season, and a negative temperature response. So we have trees that respond well, positively to precipitation and negatively to temperature. And if we then consider this one of the clusters, we do have a cluster that has a strong precipitation response to precipitation on the transition between the previous and current years. And we found 4 main clusters. So we have a strong precipitation response, a moderate precipitation response. So this is more in the pre and in the dry season. Response in the dry season, this is more just before the rainy season. And then we have a cluster of weak precipitation responses and one of negative precipitation responses. If we look at the mean climate of these areas, we can see that this cluster with the strong precipitation responses, they do have strong or drier climates, whereas these two have wetter climates. And so we can maybe divide these into drier and more humid sites. And then these subdivide again into sites with higher interannual variability in climate and lower interannual variability in climate. So it's not only about seasonality, but also how much it varies between years. And for the humid sites, we have the more seasonal and less seasonal sites responses. So in conclusion, we have these 4 main groups that have different responses, and we found also these different species can occur at the same site that have these different responses. So there's also maybe a species effect there that goes beyond these larger groups. Okay, so then we wanted to know how sensitive is growth to precipitation and temperature, and then we did multiple regressions of chronologies versus seasonal precipitation and temperature. And then we look at the wet season and dry season. And we applied then for each chronology a model selection based on a Leaps algorithm. Also a bit of a question of methodology, but it basically goes through all the possible combinations of wet season, dry season, temperature, and precipitation regressions, and chooses the most parsimonious one per chronology. And our first hypothesis was that precipitation would have in general a positive effect on growth and temperature, and negative effect. And that's basically what we found. So we have here the regression coefficients for the dry season and the wet season. And we see a positive value, the median has a positive value for precipitation and a negative value for temperature in the dry season. So if at the end of the dry season, there is some precipitation, growth responds positively. But if it's a very hot dry season, growth responds negatively. Whereas there's a positive effect in general for the wet season precipitation, but also a positive effect for wet season temperature, but it varies quite a bit. Huh? We had a second hypothesis that precipitation has a larger importance, a higher importance than temperature on the growth. So we did relative importance analysis in these multiple regressions, and indeed the blue is precipitation responses. They have a higher, significantly higher importance than precipitation, and also the dry season importance is higher than the wet season importance. So with this, we conclude that the dry season precipitation is the main driver of tropical tree growth because it has larger coefficients and also a larger importance in determining tree growth variability. So for this second part, we can conclude that we have 4 main groups of climate responses in the tropics, with precipitation in general having a positive effect and temperature having a negative effect. And the conditions in the dry season are the main driver of tropical tree growth. Okay, so we finished the second part. The third part is on tree growth responses to drought events. And this paper is almost ready to be published. At least we hope, in Science. There's quite some questions. I'm going to present two of them. And the first one is to what extent the droughts in the tropics reduce tree growth. And the second one, if these effects that we expect are negative are lagged, so they persist over time. So if we have a drought in this year, maybe the growth in the next year or the next 2 years will be also reduced. So we took all the chronologies that we have. We had 477 chronologies of these 150 species. We do have these gaps and problems with representative of climate. So we do, all of our analyses are weighted. So we do take these gaps into account. And then we took each chronology and compared it to climate data. And we took the crew data from 1930 to the present. We do analysis on seasons, so we have wet season and dry season precipitation. We remove the main trends of the climate data and then look at drought years, so the very low years of precipitation, for instance. But do it also for VPD and CWD, so vapor pressure deficit and climatic water deficit. We took then the 10 extreme years. So in this case, for wet season precipitation, the years that we are indicating here. And then we look at whether in these years, we also find strong negative responses of growth. And this is called a superposed epoch analysis. So we have an anomaly, that's the event. And we have a drought year, and then we look at whether growth has an anomaly there. So the event year compared to the mean of the remaining years. And we then selected species and areas that have at least 3 events happening. And then we have how significant and how strong are these events. So you could have a strong negative or strong reduction in growth in a year, but we also do it for the year after it and the year after it again. So we do have 2 anomalies. So for instance, growth can be reduced in the first year by 34%, let's say, and this is a significant reduction, but it could increase in the 2 years afterwards or decrease. So we do these analysis for the year of the event and the 2 years afterwards. So if you look at the tree growth anomaly for precipitation, here we have the years after drought. So year 0 is the drought year, and then 2 years afterwards. We see that there's a lot of variation in the drought responses. So in a drought year, growth is reduced, but in some species it can also increase. And in the 2 years afterwards, there's maybe not so much of a signal. So this percentage is the amount of chronologies that show a significant negative correlation. So a growth reduction. And we show also the median and bootstrapped confidence interval of all chronologies. So we see that we have a significant reduction in year 0 of about 2%, 2.5% growth. And this is true for wet season and dry season precipitation, so around 2 to 3%. In the dry season, it's a bit higher. And we see similar patterns for VPD. This is precipitation drought. This is an atmospheric drought, and this would be maybe more a cumulative drought. And we see similar patterns, a bit stronger reduction in dry season droughts, which also resonates with the previous story, but also reductions in wet season growth. So during droughts, we do get growth reductions. They are rather small, sometimes around 2, sometimes around 4%. They are similar, the responses are similar for different drought metrics. And in the dry season droughts inflict a stronger response as well. Then whether we have lagged effects, so we have a drought event here and then the 2 years afterwards, they are different. But so the drought year is significantly different from the other two, but the other two are very much close to zero. So we do have, and that's basically the same also for VPD droughts and CWD droughts as well. So after a drought event that reduces growth, growth recovers in year 1+2, which means that tree growth in the tropics is resilient to drought. So you get a disturbance, but it goes back to normal quite quickly. And it's also similar for different types of droughts. There are differences between angiosperms and gymnosperms, but I'm not going to show them here. And watch out for the paper coming out hopefully soon so that you can enjoy all these results in one go. So in short, there were 3 fronts, a quantitative review, and we showed that tropical dendrochronology is growing fast, and many areas have a good potential for dendrochronology, but there are many biases that we have to work on to get a better representation of tropical tree growth in all kinds of models. Tropical tree growth is sensitive to climate. We can identify 4 large clusters of climate responses, and dry season precipitation, especially the one on the transition between dry to wet season. So the precipitation that advances or makes the growing season starts earlier is the most important driver of tropical tree growth. And drought does reduce tropical tree growth, but these reductions are small, so tropical tree growth is resistant to drought, and they do not last for long, so tropical tree growth is also resilient to drought. And with that, I finished my presentation. We are always open for new contributions. We're going to start a new round of contributions soon to the network, so we want to expand it. We want to go towards more ecological questions, which also means that we have to change a bit the way we process the data. We need to go to the individual level. So many of the ideas of new are also we're going to start exploring as well. So if you have chronologies that are robust and about to be published or published, we are more than happy to receive them, and they will contribute for us to have a better understanding of how carbon and water works in these forests. And these are very important questions for the future. Thank you very much.
Thank you very much, Dr. Peter Groenendijk. Very interesting your presentation. Very important to know how is the advancement about dendro projects in the tropics in this region. So maybe we have any question for this presentation, please. Feel free to make any question. Okay.
So either it was everything was really clear or unclear, completely unclear, and
I have a question for you, Peter.
yes.
When you are working with tropical forests and when you find your EPS using this type of tree, how were your results about this? Why type of species achieve major values about the species population signal?
Yeah, we did not look for the network or the review to EPS. We do have some. We do look at R-bar values and correlations with mean climates. So that's for the review paper. There are some people saying that we shouldn't use EPS, that there are better metrics that we can use. That's the SSS, as I think Alan Buras is proposing that. And I think it goes a bit also towards what Neil Peterson was saying, that we have this metric and it's based on these very sensitive trees that were selected to be sensitive. And while measuring them, the ones that are not sensitive are removed from the chronologies, and then we get these fantastic EPS values in dry, cold areas, and those determine the statistics that are the metric that will be that all other trees will be judged against. And that's not fair, because if you go even in temperate regions, you can find species that have problems when we are cross-dating, that do show complacent rings. And these publications are not, these works are not published. So if you ask people, Neil Peterson, but also other people, they have we have this problematic site, we have the species does not cross-date well here, so they don't work there. They have this biased metric that the EPS should be above 0.85 or whatever, and that's just not the reality in the tropics. So it's I think we in the tropics should start a revolution maybe and say, well, it's a bit different, and trees do not behave as these long-lived pines in the high dry mountains in the US. So I don't think I responded. I answered your question, because we didn't look at EPS. Maybe we shouldn't be looking at EPS, and maybe we shouldn't be comparing it to the metrics that are imposed to us. But yeah, we have to work with annual. We have to be very careful and be sure that our rings are annual. So it's yeah, it's a bit of a contentious issue. There's a bit of a problem.
Yes.
Neil, I think you have a question.
Yeah, I'm curious. I've not worked with any tropical trees, a little bit of subtropical, but what are some of the tricks or shortcuts or ways that you use to define ring boundaries to help better see ring boundaries? We just sand. We sand a lot, but how do you approach this issue?
There are no shortcuts. That's the the main thing. So if it's a known species, you kind of do have a reference, and so for instance, Cedrela, it's a genus. Spanish cedar is a genus that's a lot used. You do get a lot of false bands, but then there are small vessels being inserted into the parenchyma band when it's a true ring, but not when it's a false ring. So those things help. Sometimes you have resin or oil canals into the ring, but not into the false rings. That's in Copaifera, for instance. But in general, I mean, ideally we would do cameo markings on a large number of species, go back 5 years later, 10 years later, sample all these markings and see whether there are 10 years out if you go back 10 years later. So that's stuff that was done in the 70s in Africa by Pierre Détienne Mariot. So they're called Mariot windows for that. So that will be an independent way of checking it. The other one is radiocarbon dating, especially we use the bomb peak, so the rise in radioactive carbon in the atmosphere. But basically, there are no shortcuts. So what we do when it's a new species, we make anatomical slices. We go to the anatomy. Sometimes it's how the radial parenchyma rays interact with the ring boundary that defines whether it's a true boundary or not. So stuff like that. Size of parenchyma, the shape of the parenchyma cells. So sometimes it's really you were saying that you need to get acquainted to your chronologies, we say that you get to get acquainted to your species, to the peculiarities of the species. You really have to learn how to read a species. And then, so my PhD students are measuring their rings, and I go there and I check them, and then I say, well, this is a mess. I wouldn't put a ring boundary here and there. And then they go, no, but this is a true boundary, this is a false boundary because of this and that and that and that. And to me, that I'm not used to the species or the genus. To me, it doesn't make a lot of sense, but to them, they can see the story, let's say. And we have radiocarbon dating, high resolution, even intraannual, showing that yes, the guy was correct. He knows his species and knows that what he's marking is a true and a false band. So and that makes the work a lot more difficult. So we can get a master thesis to evaluate maybe build one chronology for one species at one site, and that's around the limits of what is possible. So a PhD student could do maybe 3, 4. If it's an experienced student, maybe 6 or 7 chronologies, but that's about it. Whereas if you go to a temperate area that has nice rings, you just go, you collect trees, and you get nice correlations. You get automatic ring detections and everything. So you can go up to 20, 30, 40 chronologies during 4 years of research. So it's just a different type of dendrochronological science, I would say. And I think if you are planning to do a paper on talking about these changes when we go towards ecology, I think there's a lot about the tropical dendro that we can contribute to such a view, because I think that's some paradigm shifts are needed. And I think, so for instance, my PhD thesis, I have no chronologies. I am absolutely sure that my rings are not absolutely dated. So I cannot say that 1871 was a wide or narrow ring, but I know that within 5 or 10%, depending on the species, margin of error, we do have growth data. We do have ring widths. And that's for Central African forests, for which we don't have any data at all. So the contribution of a dendroecological paper there is huge. Although it's not annually resolved, although there's no climate growth correlations or whatsoever, we get a wealth of data on a forest that we know nothing about. And we can do a lot of so tree age patterns, water use efficiency changes, growth changes, all of these things. We can do that within a reasonable margin of error. So that's a bit also the boundaries of what can be done or what cannot be done with dendroecology. So it's
That's great. Thank you. And it really underscores the huge accomplishment you've all made down in the tropics. You have to go slower. It's not easy, and that's massive effort. So congratulations on that.
Yeah, and just financing as well, because if I want to go sample in the Amazon, I have to fly to either Belém or Manaus, which is 3.5 hours flight, and then I have to get a boat, maybe travel for 1 or 2 days on a boat, and then get to a site. And if we forget one thing, the whole thing stops, because you cannot go back 2 days by boat. So the logistics involved are just huge. So this pictures I'm showing here is in the upper Rio Negro, where you can either get by boat or by chartering a plane. So if you do get some data there, it's a lot more valuable than some new Quercus or Fagus or whatever data that can be produced. And although the quality is lower and the questions you can ask are maybe less diverse, it's still really interesting and relevant data.
Agreed.
Okay, very interesting your presentation there, Dr. Peter. The magnitude of this species that you are using for Dendro project is so important for Dendro community and our work. Thank you so much.